So the talk that I want to give today has two parts. The first part, I will just briefly describe gene editing, which is synonymous with genome editing. Gene editing should be understood as an umbrella term for a basket of techniques. Most involve some kind of targeted cutting system using an enzyme that cuts DNA and which is inserted into living cells. These living cells may be animal or plant or other cells, and although the initial alteration occurs in just one cell, it is important to remember that this one cell becomes the basis for a crop or animal breeding program. The exception to this description is a method called oligo-directed mutagenesis, or ODM. ODM is a technique developed by a company called Cebus, which is based in San Diego. ODM is often referred to as a form of gene editing, but in practice it has virtually nothing in common with other forms of gene editing, since the method involves inserting small stretches of DNA into cells. So no DNA cutting enzyme is used in this case. So it's important to appreciate in the context of this talk that each of these different gene editing techniques likely has a different risk profile in terms of the genetic damage it causes to the genome of whatever cell it is inserted into. It is also important to appreciate that virtually every laboratory or company uses these techniques differently, either slightly or very differently sometimes, or they use different versions of CRISPR, for example, or they use it on different species or at different concentrations, or targets different genome sequences. So in all probability, these differences affect the risk profile of the technique in terms of the genetic damage caused to the genome. So what I'm saying here is that people are publishing papers describing some of these off-target effects and the various consequences, but we have to understand these as kind of token examples, if you like. So some species methods or genome targets may be much more prone to genetic damage than others or much less. The important fact to keep in mind is that we really have no idea. So when people talk about rolling out gene editing techniques for specialty crops or hard to engineer crops, which they do, these people are venturing far into uncharted realms when it comes to biosafety. There is probably a huge amount of devil in these seeming details, but the bottom line is that we do not know how much these seeming details matter. An important relating issue, and especially this is true for company developers, is that they're not at all transparent about their methods for developing new crops, which they call gene editing. So a case in point is Cebus with its ODM method. This company recently applied to the European Union for approval of the first gene edited crop. And then as the crop began to hit regulatory buffers, Cebus changed its mind and told the EU that its canola was not gene edited after all. It had arisen by chance, they said. So on this basis, and I think this is fairly predictable, transparency is going to be a major issue. Who did what in what crop and so forth. And in the second part of the talk, I'll talk about the steady emergence of reports of genetic damage associated with gene editing. We now know, for example, that there can be major genetic damage and inadvertently introduced DNA sequences at the target site. More slowly, we are learning that there can be genetic damage all across the genome, which can arise for a variety of reasons. And the last thing I want to note is that the word editing itself in this slide is in inverted commas, and I'll use those inverted commas throughout the talk. So these inverted commas emphasize that the term editing is a marketing term. It conceals more than it reveals in that it implies micro changes to the genome and also some similarity with editing on a word processor, which of course is very precise. But the important difference is not only that genome manipulation has nothing in common with word processing kinds of editing, it is that the term editing was invented in advance of anyone collecting evidence for what actually happens inside cells which are edited. So this slide shows you some of the different types of gene editing methods. So as I said, it's a basket of different techniques, and all of them have in common, except for the ODM method, that there is an enzyme that cuts the DNA, and there's a targeting system which brings the enzyme to the place where it's supposed to cut the DNA. This is the theory of the whole thing. And so each one of these methods, the meganuclease, so the acronyms are explained down at the bottom left here. But in each of these cases, there is a protein that cuts and a protein that guides the cutting enzyme to the piece of DNA, to the right piece of DNA. The exception to this is CRISPR. So CRISPR is the bottom right technology, and it uses as its targeting system an RNA molecule instead of a protein molecule. And so this is a small difference, and I'm going to focus on CRISPR for the rest of this talk. And that's because there's most research about CRISPR and its off-target effects and genetic consequences, but also because it seems it's the one that's most used by researchers. It's not necessarily the one that's closest to the market or most used commercially, and that is more of an open question. And we still don't know which kind of technology will win out, as it were, in terms of market forces. But I'm going to focus on CRISPR. And as I said, it has two components. It has a protein component, which is the Cas9 protein. So it's a nuclease. So a nuclease is an enzyme that cuts DNA. And the CRISPR is the RNA component of that. So what a researcher does is they produce an RNA component, part of which is always the same. And then the green part, which is, I should be able to use this little arrow. The green part here is different every time, and that targets the molecule to the genome, the cutting enzyme. So here is a picture of it a little up close, somebody else's demonstration of how it works. But essentially what you can see here is you have an RNA sequence, which is the gRNA, this part here. And what it does is it basically inserts between two strands of DNA. And when it finds a match that it likes, it cuts the DNA in two, right? And what you have to understand about a DNA cut is it's not an edit, right? An edit would be a replacement of an A with a T or a G with a C, for example, in the genome sequence. But that's not what's happening here. What's happening, and the same is true for all the others, is that simply the enzyme is making a cut. And then what happens once that enzyme is removed and disappears, the cut has to be repaired by the cell. And what the researcher has to do is try to select repair events. So they will basically do this in thousands and tens of thousands, sometimes of cells. They have to select a repair event that suits their needs. So the cell will repair these events, and it does so fairly at random. But it also does it in a kind of a panic mode, because even though this looks like a small change to the DNA, you've just cut it, surely it can just recombine. What you're doing here is you're cutting a chromosome in half, and the chromosome has the potential to when the chromosome is cut in half, that means the cell can no longer participate in cell division. That cell is basically at a dead end because it can't transfer its genome into the next generation in a rigorous kind of logical way, because basically the genome becomes fragmented. So the cell has to repair that sequence, that cut. And so it does so often in kind of a panic mode. So what is emerging from the off-target research is basically that all kinds of strange DNA can be added at the cut site, which is not consistent with a gene editing type description. So one variant of this method is that the researcher can supply their own DNA, which the cell can use to repair the cut. And so what the researcher would then do is basically do the gene editing process, but they would select for repairs that use the double-stranded DNA that they themselves have supplied. So once again, this is kind of a deviation from the whole editing idea, in the sense that one can actually introduce, major transgenes and antibiotic resistance genes, or herbicide tolerance genes. There's all kinds of traits that potentially can be added that companies are hoping they can just get by calling these gene editing. So this is kind of the state of the market, at least in the US, of gene editing. From probably a year ago, I made this slide. So I just wanted to give you a kind of overview of where we're at. So in this first example, Penn State University has made a non-browning mushroom, and they published one paper, and there's no sign that will ever come to the market. And then the other examples are made by companies. And the reason I put boxes with question marks inside them is that we don't even know enough about them to have photographs available. So practically no information is available about these. And then in the case of Cibus's canola, they consider they're very close to the market. But as I said, there's been some interesting things going on with their approval process. Right? They basically changed their minds that their crop is a gene edited crop. And this underlines the whole issue of transparency. No one can really know what the company has done to its crop if they don't honestly tell us. So I just want to go through some of the research about the kinds of imprecision that are associated with gene editing. So this was a paper that was published a few years back, I think it was 2016, and it's kind of kick-started the whole idea that gene editing might actually, despite its name, not be very precise after all. So there were some papers published before this, but the research community kind of felt that they could ignore that because the authors of the research were not famous, and there were calls for their papers to be retracted as they couldn't possibly be true, and so on and so forth. But eventually, with the publication of this paper, it became acceptable in the scientific literature to argue that there was imprecision at gene editing sites. And what these people showed is that the gene edited cell is basically repairing the breaks produced by the gene editing mechanism by leading to either large losses of DNA or to complex rearrangements of chromosomes, which are also highly problematic events for the cell. And so they're likely to lead to basically mutant plants. So, and then there's the whole issue of how does a researcher actually make the edited crop in the first place? So one of the key issues that nobody seems to be paying too much attention to is the fact that in order to make an edited cell, you have to introduce the editing apparatus into that cell. And the way that most people have been doing this is by basically shooting DNA, introducing DNA into the edited cells, which codes for the enzymes or the RNA or the targeting protein that does the editing process. But what that means is that you have to introduce a lot of foreign DNA into those cells. So you can't just add the DNA that codes for the CRISPR and the Cas9. You also have to add all these ancillary parts, which potentially can end up in the genome, very likely, in fact, can end up in the genome of the edited crop. So instantly we have a problem here in that the edited crop is going to be potentially have all this foreign DNA, which comes from all over the biological world, put into it. So this is an issue of the source of the DNA. Then the next question is, you have to prepare, and I'm giving an example here of plant cells, but similar comments could be made about animal cell editing. Is that you have to prepare the plant cells to receive the DNA. So you're going to shoot the DNA. You're going to use basically a modified gun to shoot the DNA on pellets into the cells. And you have to get the cells in a fit state to receive that DNA. And the way that you do that is basically put them through a thing called tissue culture, which is very mutagenic, right? So thousands probably of mutations in each cell arise as a consequence of this tissue culture. So, you know, one of the ironies of all this is that the editing process is supposed to be very, very precise, but the developers are putting their cells through extremely challenging situations. And likely the number of mutations that will be introduced by the editing process will be dwarfed by the number of mutations introduced by the kind of procedures, what you might call the ancillary procedures that go along with this. So let me just show you an example of this, right? So imagine what happens when you want to shoot a pellet. So you've got I've got it drawn this as a red pellet with some orangey colored DNA on it. Essentially, you put the DNA on the pellet, and then you're going to shoot that using a gun into the cell. When you do that, the pellet basically has to pass through half of the cell before it gets to the nucleus. It may pass through a chloroplast or a mitochondrion, some cellular organ. And when it passes through those organs, it collects DNA. They're also full of DNA. So it will collect pieces of DNA. It will lose pieces of DNA. It will finally lodge in the nucleus. And in order for the DNA that you've added to insert into the nucleus, you first have to damage the DNA. So the pellet is basically crashing into the chromosomes in the nucleus and damaging them. Because, and we know this because if you don't damage the DNA, then there's no reason for the cell to take up the DNA introduced in the pellet. So you've got this very damaging process of shooting DNA in pellets into the nucleus that also results in major genetic damage. So this isn't the only method of doing it, but it is the most popular one. And so I'm just focusing on that really as an example.
Jonathan, if I can request you to wrap up in a few minutes, couple of minutes.
Yeah, we're nearly there, Ruchi, thanks. So the third kind of genetic damage that interests me, and you know, what I'm doing here is picking and choosing bits from all over, but essentially simply because there's not time in this talk to talk about all the different kinds of genetic damage. But this is a really interesting one. So this is a study done last year where researchers basically took, they edited some mouse cells, and they took those mouse cells and they then sequenced the DNA of the insertion sites. And what they discovered is that they had inadvertently added DNA from E. coli, which is a bacterium, DNA from a cow, which is bovine, and they'd also introduced DNA from goats. And they scratched their heads for quite a while, like, where does all this DNA come from that we didn't intend to put into these cells? And it's now ended up in these cells at the editing site and at different places in the genome. And it turned out that they're contaminants of the process used. So basically, as I said, they're taking DNA and they're having to purify some DNA in order to shoot the editing proteins shoot the editing mechanism into the cell, but that DNA has contaminants in it. And the contaminant in this case was E. coli because they'd grown up the DNA in an E. coli cell. And the bovine DNA and the goat DNA came from other forms of contamination that were basically requirements of cell culture, right? So cell culture requires purified proteins, and those purified proteins come from cells that contain DNA. And if you use bovine DNA or goat DNA, that is where your contamination comes from. So this is completely unexpected, and it is totally inconsistent with the whole idea of genome editing. So this is really a summary slide. So we're just about at the end, Ruchi. So what I want to really say is that genetic consequences stem from the manipulation of cell cultures and the need to introduce the gene editing components into cells, right? So this is basically all the ancillary stuff that you need to do your editing in the first place. And the damage to the genome is essentially random. It's not going to occur at your edited site. And then there's another set of genetic consequences which stem from the functioning of the edited gene editing machinery itself. So the gene editing machinery can cut the DNA on the target, but it can also mistakenly cut other places in the genome, which people call off-target cuts. And I haven't even had time to talk about those, but basically it's not a guarantee that the editing machinery will only cut where you want it to. And all of these, if you add up all these issues, what they basically do is they say, this places a very high premium on the diligence and competence of the breeder, right? Because all these changes can happen, but it's up to them to make sure that those don't find their way into the final product, that they exclude ones, that they actually even know how to detect these changes in the first place. And at the moment, I don't know how many people have followed the hornless cow story, in which basically a company who was pioneering cow breeding with breeding hornless cows, they basically inadvertently introduced an antibiotic resistance gene into their cows. And they had no idea it was there. It was perfectly obvious. Just a simple piece of sequencing from the FDA detected it, but the company itself didn't do that, right? So there's a huge question over the diligence and competence of all these startup companies and the multinationals that are actually doing this. So I'm going to end there, and I'm happy to take questions if there's time, but that's up to you, Ruchi. So thanks very much, everyone. Oh, please subscribe to our website. Given the way the internet is these days, it's going to be difficult for you to keep up with all this information if you don't subscribe to us. So thank you. There's a company, Cebus. There's also another company called Calyxt. But there's also, one thing that's interesting about all this is that there are some, what you tend to hear about are very small companies. These are kind of startups. But the real people behind this who really want to use this is Dow and Bayer and the bigger companies, right? They're kind of waiting in the wings, using these small companies as front men, if you like. And so, we should know the names of these little companies, but we should also understand that it's the big companies that really are behind all this. They have the lobbying power to make the governments change regulations and to deregulate GMOs and so forth. This is not a technology that's going to be controlled by the little guy. And so you also hear people talk a lot about how this will be done in garages and so forth, right? There's no chance that companies that set up in garages are going to control the food supply, right? Or offer important products for the food supply, because these people don't have the marketing now, they don't have the testing now, they don't have the breeding, they don't have the breeding contacts and so forth, right? This is not going to happen in garages. This whole idea of startups in garages is just a front that's put up for the benefit of the media and for the benefit of populations. So I can share some contacts, but for the most part, the companies that are involved is Dow and Bayer and the real big companies. But the most useful thing I can offer really is to go to our Independent Science News website and look at some of the articles. The articles are not too long and there are not too many, but they kind of summarize a lot of the issues of gene editing in terms of genetic damage, but also in terms of the paradigm that Vandana was talking about. The idea that we published an article, The 3 Myths of Gene Editing, which is basically about the idea that if organisms are self-organizing systems, whether they will be amenable to gene editing or not, whether they're amenable, what does this have to do with the failure of GMOs as a concept, right? We've seen the history of GMOs has been the companies have this tiny limited number of products, right? They have their herbicide tolerance and they have their insect resistance, but they never seem to come up with the, no reliance on fertilizers, no reliance, the resistance to climate change. They never come up with the complex traits, right? And it has to do with the conceptual flaw that the companies have, which is that they have a genetic determinist mindset, and that mindset basically is a road to nowhere. And so this is the fundamental issue here.